Thank you, Heidi, and thank you all for coming. Great to see such a great turnout. It's a real honor to be here. In thinking about the possibilities I would have, I can't think of anything that is a real more of an honor and such a great humbling opportunity than this one, thinking of those who have been before me in the Discovery Lecture series. And then for me, Vanderbilt is such a special place, as Heidi said, going back to my early days. And I think when you get your training at a place and then you come back, you still have this sense of being a trainee and just the tremendous respect for so many people here. But I'm going to talk to you a bit today. One thing that I also wanted to just mention is thank you to the artist who drew this fabulous picture of me. I've seen it several times now, and it's the me that I've always wanted to be. So I finally have arrived at that perfect look. But thank you very much for that. So I'm going to spend the next hour or so talking to you first in a very big picture way about studies that have been done over the decades that are relevant to drug discovery, and really more talking about drug discovery in general from a big picture. What are some of the challenges and issues where we are today in drug discovery, and then give you a couple of examples from our own of how we're trying to have an impact in the drug discovery arena. So in thinking about the advances that have occurred over the last couple of decades, I think all of us in the biomedical sciences have been extremely excited to see tremendous advances in a number of areas. We now have an understanding of the basic biological mechanisms involved in controlling a wide range of animal functions, both normal physiology and pathological functions. We have an understanding of human disease that I think is unparalleled in terms of thinking about human disease at a molecular level. So now when we think about Alzheimer's disease, many issues that were really unknown only 10 years ago, we now have a much better understanding. The same can be said for diabetes, for various cancers, where our knowledge base has increased tremendously. There's also been major advances, such as the one that is kind of illustrated here, with such as the cloning of the human genome. This was a very widely celebrated achievement that occurred at the first part of this decade and this millennium, in which the entire sequence of the human genome was determined and reported, and has generated tremendous excitement and also a number of tools that we as biomedical sciences, in the biomedical sciences, can use in our research. So we see advances such as this, other advances in automated technologies, high throughput approaches to chemistry, all of the things that are really critical for drug discovery, and it gives us tremendous optimism about the opportunities that we have for developing new medicines. The issue that I think concerns many of us is we look at advances such as this and the others that I've just alluded to, and then we also look at the advances in novel therapeutics actually making it to market, and there's a major disconnect that occurs, so that we see tremendous advances in the biomedical sciences and less translation of those advances to new medicines. So that when we look over the last few years, there has been a steady decline in the number of new medicines that have been approved by the FDA that become available for clinical use. Just to take a snapshot in 2002, looking at the Orange Book put out by the FDA, in that year there were just over 10,000 drugs that were approved for use, reported in the Orange Book. But when you look at these drugs, you find that while this looks like a tremendous number, that it actually only represents 433 distinct chemical entities. So while it looks like a large number of medicines that are available for clinicians in treating their patients, that really represents a relatively small number of distinct chemical entities or medicines. The reason is that many of these 10,000 drugs are combinations of different molecules, such as acetaminophen. So acetaminophen is present in 337 of those compounds, and so there are a lot of different mixtures that make up this number. And also, when you look at these 433 new chemical entities, many of these were drugs that were approved well before this revolution that we've seen in biomedical science. So in 1938, many of the compounds that we now have available were already available. And then the compounds that have been developed more recently, many of those are piggyback compounds that are slight modifications of drugs that were previously available. So when we think about this, I think it's critical that we all think about why that is the case and what we can do to start to really realize the opportunities that we have in biomedical sciences. And I think it's very helpful to go back and look at the history of drug discovery and consider how things have changed and how our paradigms have changed and how this may influence this disconnect. When we look at classical drug discovery paradigms that are responsible for generation of many of the drugs or most of the drugs that we have available today, this goes back to medicines that were actually in use for many centuries or even millennia in the form of different medicinal plants. And the approach to drug discovery in these early days was with the advent of the modern approaches to purification of compounds from plant extracts, synthesis. We were able to take these known medicinally active plants, isolate active compounds from those, determine the structure of those compounds, then once we've determined that structure, prove the biological activity of the principal agent rather than the plant itself. And from there, we know we have the medicinally relevant agent in that extract, and we can use that as a starting place to begin to make improvements by chemically modifying the structure. So in this paradigm, we go into the process with an absolute assurance that we will see clinical efficacy because these are compounds that have been used by men for long periods of time, and that there's not a serious toxicity that is going to limit their use. So the challenge is relatively simple of identifying that key component and then making improvements on it. There's no requirement in this model for an understanding of the molecular target or the cellular actions of that molecular target. What is really important is just knowing this clinical efficacy. So this dramatically impacts the way that drug discovery was approached. And as the drug industry, the pharmaceutical industry began to emerge, it was emerging in the context of this technology. And this is a list that I don't need to show to this audience because we are all familiar with these drugs and we all know their origins. But just to remind you of some of the really important medicines that are still available and are commonly used today. Morphine, one of the mainstays for pain treatment, or the mainstay still today, has been in use for many millennia, dates back to 3400 BC from the opium poppy. Digitalis is obtained from foxglove plant, was first referred to in the 1500s for its therapeutic use. Aspirin was first referred to as, or salicylic acid, the component of willow tree bark that is the deacetylated form of aspirin, was first discussed by Hippocrates in 400 BC. Novocaine was derived from cocaine, the first local anesthetic. And the list goes on in terms of the numbers of medicines that we still use today, still rely on, that came from this classical paradigm. There have been some changes that occurred during the first half of the last century, the 1950s and 60s and before that, in which a number of drugs were discovered that were not derived from medicinal plants, but still followed a similar paradigm in that there was a known efficacy. And a good example of this in my field is chlorpromazine, which is an antihistamine that was being used in patients suffering from schizophrenia. And what the very astute clinicians began to notice is that in addition to the antihistamine properties, and actually they were using this for the sedative properties of some antihistamines, that chlorpromazine also had true antipsychotic efficacy, reducing hallucinations, delusions, thought disorder in these patients. So from those very astute clinical observations, it was appreciated that these are antipsychotic agents, And they immediately began to go into clinical use as antipsychotics, but this also initiated a major effort to discover the mechanisms of antipsychotic action, ultimately leading to the discovery that these compounds act on D2 dopamine receptors, and then developing new programs aimed at identifying novel compounds that act on the dopamine receptors but do not have the antihistamine and other undesirable properties. So once again, in this paradigm, you go into the process with an assurance of clinical efficacy in that chlorpromazine had established efficacy. Once it was clear that the target for chlorpromazine was D2 dopamine receptors, there was a high level of confidence that development of new compounds with that activity would have clinical efficacy. So there was a heavy reliance on first medicinal plants and then serendipity for discovery of new medicines, and this has been tremendous, and I think still needs to be a major part of how we think about developing new drugs. But exclusive reliance on these old paradigms will not lead to the types of breakthrough medications that we all would like to see. So when we look at some of the diseases that we are dealing with today, there are no medicinal plants that are obvious in many cases. We really need to understand the biology of the disorders and begin to develop new medicines in that way. So there was a major shift that occurred in the way that drug discovery is done that really began, I think, one of the major projects that I think led to this shift was the discovery of cimetidine, or Tagamet, which is an H2 histamine receptor antagonist. And in this new paradigm, there's now an effort to understand the biology of a disorder, identify molecular targets that may alter that biology. From identifying those targets, then develop a drug that acts on that target, and then after this is complete and you've developed a molecule that is safe and useful in man, or one that you can use for hypothesis testing in man, you then go into clinical trials in which you test the hypothesis that the drug will have clinical efficacy. And that last statement is the key statement that I think fundamentally sets apart what we often try to do today from the old paradigms, in that we now spend a tremendous effort to get to the point where we can test this hypothesis. And this, as I mentioned, began with discovery of cimetidine. When I was a graduate student here at Vanderbilt, it was just after cimetidine had been launched. It was launched in 1976, and I still well remember the lectures on the discovery of cimetidine. It was a very exciting time, and I remember sitting in classes actually in this room in pharmacology, listening to the lectures and being inspired by the power of this new approach. So cimetidine was discovered as an H2 histamine receptor antagonist, but this also was a relatively simple program when you look at it compared to many of today's programs, in that the starting point was histamine itself, and then a relatively small number of molecules developed trying to develop compounds that bind to the histamine site but do not activate the receptor and selectively block the H2 receptor. So now we have entered fully into the era of using this paradigm for drug discovery. And I really think that this breakthrough with cimetidine, others that occurred near the same time in the same vein, led to a fundamental shift in the way many of us in the biomedical sciences approach our work and think. And that is that we are less impacted oftentimes by thinking about the clinical data that we're seeing with existing drugs, and we're more impacted by thinking of mechanisms, cellular mechanisms, biological mechanisms, with the idea that we will then follow this approach and test a hypothesis in the clinic. So using today's paradigms, we now identify targets. There are now at least 20,000 genes in the human genome that give us a tremendous number of potential targets to choose from. We think about those genes that are expressed, how they impact human biology and animal biology, and then identify targets that we think may have potential as drug targets. Once those targets are identified, we then go through the process of doing experiments to try to validate the targets, get some information as to whether they will be useful as drugs. Once we make a decision to pursue a target as a drug target, we then have to initiate a process of identifying leads, because we're no longer working in a setting where we have agents that are already present in plants or in drugs from which serendipitous discoveries have been made. So we need to find entirely new molecules that will interact with the target that we're interested in. And to do this, we typically run high-throughput screens looking at large numbers of molecules that have drug-like properties to try to identify compounds that have activity at the targets that we are interested in. We then initiate a major effort in optimizing those initial hits, and this is where the real muscle and expense of drug discovery begins in many ways. And that is when the chemistry effort is initiated to try to take this initial hits to the point where it can go into clinical development to ultimately test that hypothesis. And in modern drug discovery settings, this is usually done by a team of about 30 to 60 scientists, maybe 15 to 20 chemists on any given project, along with all the people doing drug metabolism, pharmacokinetics, the other aspects of biology. And this large team of scientists may work for 2 to 3 years before they come up with a compound that's ready to be tested for toxicity in animal models first, and then ultimately go into clinical development. This is an expensive enterprise that costs about $40 to $50 million just for this step of the drug discovery process typically. And then from there, you go into process chemistry, a very different type of chemistry to make large quantities that can be distributed. Most companies will do this right up front so that before they go into development, they want to make certain that they've already established the methods for generating multi-kilogram quantities that can be distributed worldwide, and then ultimately preclinical and clinical development. So when you look at this process, it's a very lengthy process. The clinical development side takes approximately 7 years. The total timeline is on the order of 12 to 14 years. And you think of that in relation to the patent life of an agent, which is 20 years. So each year that you move along this pathway, you're eating up that time in terms of the patent life of a possible drug. When you move into this development phase, the other thing that is critical to keep in mind, the output of that discovery phase, which in the previous slide was outlined in the red box, is a development candidate. And this development candidate then goes into preclinical development, so toxicity testing in animals, then into phase 1, phase 2, and phase 3 clinical development. This shows the timelines for those different phases of the development process, but also the attrition rate of compounds in this process. So after this massive investment in drug discovery, only 10 of 50 compounds that go into preclinical development actually make it through so that they can go into phase 1. Only 3 of 10 that go into phase 1, and ultimately only 1 out of 50 compounds that goes into this advanced stage make it to the market. So you have a tremendous attrition rate coupled with these long timelines that are critical for drug development. When you see this in terms of the cost, what you see is a tremendous cost of discovery of a novel drug. And I think we've all heard these numbers many times. We've heard them grow over recent years. But now the estimated cost of taking a drug from initial target identification to market is about $1.2 billion. So a massive expense for discovery of a new therapeutic agent. And you can see that that expense increases exponentially, especially as you move into the clinical development phase. And the key thing to keep in mind with this new paradigm for drug discovery and development is at the end of this massive cost and spending is we have come to a point not where we have a new medicine in most programs, but to a point where we can test a hypothesis. And all of us in biomedical sciences know that very few of our hypotheses are correct the first time around. I have rarely in my own case seen a situation where I had an idea that I thought was brilliant, and it actually turned out to be brilliant, because biology always tells me something different. And so when we get to that end stage, after this massive effort, we're testing a hypothesis. In all likelihood, it is not going to be entirely correct. In the science at the level that most of us work, we go back and refine the hypothesis, do additional experiments, but that's extremely difficult in this context because of the cost. So you can see this escalating cost that has gone from approximately $200 million to $1.2 billion per drug since 1980. The total spending worldwide for drug discovery and development in the pharmaceutical industry has gone from $2 billion in 1980 to $32 billion today. So there's been a tremendous increase in spending for drug discovery and development. Despite this tremendous increase, the question that we're left with is, where are the breakthroughs that should come from that and that we all anticipate and are hoping for? The problem that we have is that discovery of novel breakthrough medicines, it requires this massive investment for testing of a highly speculative hypothesis. So once again, we go through this process, but then we ultimately get to the clinic to do an experiment. And if we get to the clinic and find out that there is no efficacy, or that there are other problems that are associated with that drug, then obviously that is not going to make it to market. So there's a critical need for us to change the paradigms that we use for drug discovery and development, to think innovatively of new ways in which we can approach this that will give us a higher likelihood of success and reduce the risk so that we are able to capitalize on the tremendous opportunities that we referred to earlier. So we need to do this, however, without sacrificing innovation, or we'll never capitalize on those opportunities. And there are a number of ways that companies are reducing risk that do not lead to innovation, such as Me Too drugs, slight improvement of existing drugs. And those are things that allow this machine to continue to operate, but ultimately do not lead to the breakthroughs that we all hope for. So I think we as a biomedical research community need to put a lot of time into thinking about this problem. How can we begin to shift the paradigms for drug discovery in an academic institution? How can we as academic scientists contribute to this process so that the entire enterprise of biomedical research is able to deliver the societal gains and health gains that we all hope for? So for me, I am most focused in neuroscience. I'm a neuropharmacologist, and my interest is largely in developing new strategies for treatment of psychiatric and neurological disorders. And so I think about the issues that lead to this attrition and the loss of compounds, the failure of compounds, and for CNS active drugs, for psychiatric and neurological disorders, it turns out that 70% of failures of drugs are due to a lack of efficacy that is seen in the clinic. So when you have a failure due to lack of efficacy, the problem with that is that with the current paradigms, we don't recognize that lack of efficacy until phase 2, late phase 2, or even phase 3. So at the very end of this very expensive campaign, and then we realize that the efficacy is not there. This percentage varies for different indications. It happens to be very similar for oncology and cancer, so it's very difficult to predict based on our current models those approaches that are going to have efficacy in the clinic. For antibiotics, antimicrobial agents, toxicity tends to be the reason that most compounds fail, because there we have very good models in terms of ability of a drug to kill a microorganism if it is safe. But once again, thinking about neuroscience, where most of the failures are due to a failure of efficacy, I think it's very important to think about ways that we can improve this. And there are a number of ways that we can approach this, including thinking about better models of predicting efficacy based on animal models, etc. Also, I think the toxicity end is critical, thinking of better predictors of toxicity, better early clinical predictors of toxicity. But for now, what I want to focus on is just thinking about how we can change the way that we are approaching our science in many cases, and I'll speak for the neurosciences, so that we now return to a bedside to bench approach for thinking about therapeutic discovery. And when I say this, I think many of us in this room already use a bedside to bench approach, and that is we see observations in the clinic, we think about those observations, we take those back to the laboratory and try to use those to make advances in new therapeutics. But I think during the time that I was a graduate student here, that was the mainstay of neuropharmacology. So we talked a lot about chlorpromazine and other antipsychotics that had initially been identified serendipitously. There was a major emphasis of academic scientists on determining the mechanisms of action of those compounds. The same with the antidepressants, so that anytime you saw clinical efficacy, the biomedical research community in academic institutions, NIH-funded research, would be heavily focused on understanding those mechanisms so that we could realize these advances. Today, there is much less of a focus on that because we oftentimes start with the cell and the molecular systems we're interested in. We then move from that direction. And all of that is critical and important, but I think that a component of what we should do is to return to this bedside to bench approach. There are several ways to do this, and just listed here, and then I'll give examples of a couple. First is discovering mechanisms of known therapeutic agents for which the mechanism is not known. There are many examples of that. One that stands out to me right now is a compound modafinil, Provigil, which is used for treatment of sleep disorders. The mechanism of action is unknown. It is a very big success in the clinic, used widely, and yet we really do not understand the mechanism. When I first returned to academics, I was on an NIMH panel to look at the direction of NIMH, evaluate all of the grants that were being funded, and come up with ideas about how the NIMH could start to have more of an impact in terms of therapeutics. And in that process, went through the abstracts of every grant in that institute, and I found that there was only one grant funded that included modafinil at any level, and it was completely non-mechanistic. It was a grant that was funding a clinical study that was not at all focused on determining mechanism. I was actually shocked to find that there was very little effort at identifying the mechanism of modafinil. Another example that's been a real success is a compound phenibam that was found to have anxiolytic activity in 1982 with no known mechanism of action, developed by Roche. It ran into trouble in clinical development due to toxicity and pharmacokinetics. But they stopped the program without a known mechanism of action. And just within the last 3 years, it's been found that the mechanism of action of phenibam is inhibition of one of the metabotropic glutamate receptors that Heidi mentioned, mGluR5. And so this is an example of a real success of taking a compound with known efficacy, now understanding the mechanism so that we can develop new molecules that have that same activity and hopefully lead to the same efficacy. So that's one approach, and this is an approach that I think is happening every day here at Vanderbilt. I think of some of the COX-2 inhibitor studies, some of the actions of COX-2 inhibitors, like the work that people like John Oates and Larry Marnett are doing in both cancer and CNS, and trying to understand these actions that have been identified that may not be related to COX-2 inhibition, but some other target, and there are many, many other examples of that. And I would just encourage us to continue to look for these opportunities and focus on them. Another is an aggressive focus on development and clinical testing of proof of concept compounds. And this is something that in the older models, the recent models, I should say, in the pharmaceutical industry has not been really pursued, in that most companies would go through the same thinking that they went through back in the days when they were almost assured of efficacy. They would go through a drug discovery and development program, and before they would go into any clinical efficacy testing, they would wait until they had a compound that was suitable for once daily dosing, perfect pharmacokinetics, all the properties needed to make it as a successful compound on the market. They would then go through all the process chemistry that was really not needed if it never did go to market. And in most of those cases, the drug did not make it to market. One thing that I think is critical is to begin to focus on development of compounds that are safe well before a drug that would actually make it to market that allow proof of concept studies in the clinic, and then going in with very specific hypothesis testing, experimental medicine studies to determine whether those compounds may have efficacy, and then if they do, return to developing a compound that can go through the entire development process. There are examples of this recently that have been very exciting and to me encouraging. One is a Lilly compound that really does not have the properties to go the distance, never has been viewed as that, I don't think, but was shown to have efficacy in treatment of anxiety disorders, and now major efforts on developing better molecules that can be marketed for that indication. A CGRP antagonist shown to have efficacy in migraine, but only available IV. But because of that proof of concept, we now know that this is an effective approach, and we can move forward in developing new medicines. Then two approaches that I just want to touch on and give examples from our own work are building new understanding from clinical studies that do not necessarily involve drugs, but other clinical observations that give insights for new approaches to developing drugs for treatment, and then clinical experience from serendipitous discoveries with drugs in the clinic that I'll mention after that. So for this first example, this is a program that we've been involved in for many years now, focused on Parkinson's disease. And this began when I was at Emory University and interacting very closely with a terrific group of neurologists there who spent their lives thinking about the basal ganglia circuit and how it's involved in controlling motor function and how basal ganglia function is altered in Parkinson's disease, and then from that developing new therapeutic strategies with surgeries for treatment of Parkinson's patients. So this was very well done combination of basic and clinical research, recording from patients during the process of surgery to understand firing patterns in different nuclei in the circuit that controls movement, and also doing the same studies in non-human primates in parallel, and then developing an understanding that ultimately led to this model. And I'm not going to go into any detail about this, but just want to mention that these large arrows represent synaptic projections where activity is dramatically increased in Parkinson's patients. And if you can correct this increase in ultimately excitatory transmission at this synapse, but through these other changes, this has a tremendous therapeutic benefit. And this was done, has been validated clinically through surgical techniques. I'll just show a video. This is small. I hope that people in the back can see it. I'm not sure if I have a way of increasing the size. Let me start over. I won't try to increase it, but hopefully you can all see it. This is a patient at Emory when I was there in 1998, and she has very advanced Parkinson's disease. You're now seeing her before surgery, and this is a patient who is now maximally effectively treated with L-DOPA. She's in an off state. She is completely immobile. The nurse was asking her to stand. She was unable to stand without help. You see tremor in her hand. And now the nurse is asking her if she can walk across the room, and she's completely unable to walk unaided. So very severe advanced Parkinson's disease. Now what you see is after a unilateral surgery that inactivates the STN, this can be done either with lesions or high-frequency stimulation. And now after this unilateral surgery, you see that she was able to stand alone. She's able to walk across the room, a tremendous gain of mobility. This is Jerry Vitek, a neurologist there at Emory that was a close colleague that really pioneered many of these studies. And what you'll see next is the same patient after going back in and doing the bilateral surgery. And so this is the same patient who is really at the end of the road in terms of effective treatment for Parkinson's disease, has really experienced a new life. And this surgery is occurring here at Vanderbilt. Many of you in this audience know it well. But a very exciting advance in terms of therapeutics. But I would contend that also, and perhaps ultimately more importantly, a very exciting advance in terms of validation of this model. And what it tells us is that if we can reduce transmission through this component of this circuit, we can indeed have a therapeutic effect. So based on this, we felt that this provided a strong clinical basis for moving forward in a concerted effort to identify drug targets that are present within the nuclei of the basal ganglia that may be druggable targets and that could have activity that would allow the same effect to be accomplished, but with pharmacological approach rather than a surgical approach. So we began a large effort that included, we were primarily using non-human primates at this time because it allowed larger amounts of tissue from these different nuclei, and we started profiling all of the major nuclei in this circuit, looking for genes that would express druggable targets. And this included a variety of different approaches, including basic gene arrays, but also more low throughput approaches such as northern blots and PCR Tagamet analysis, looking for genes that we thought could be relevant. And really it was the low abundance genes that ended up being the most interesting, and therefore genes that came up through the lower throughput approaches. And one of those was a gene for a receptor called mGluR4, a metabotropic glutamate receptor that Heidi referred to, that we found was expressed at high levels in the striatum. That's all we knew at that point, is that there was high mRNA expression. But what we also knew is that closely related members of this receptor family in other regions were trafficked to presynaptic terminals from their cells of origin, where they regulated transmitter release. So we made the effort to develop specific antibodies for mGluR4. We developed highly specific antibodies, used those for a detailed anatomical analysis, and through that found that mGluR4 is localized in presynaptic terminals at this first synapse in this circuit, the striatopalatal synapse, that is thought to be overactive in Parkinson's disease. After a number of studies in which we clearly established that mGluR4 was present at this synapse, we then asked the question of what effect activation of this receptor would have in these presynaptic terminals. And to do that, we made slices of the midbrain in which we were able to maintain this component of the circuit intact in vitro, and used patch clamp recordings in these slices to record activity at this synapse, and then added an agonist of mGluR4 and found that it completely shut down neurotransmitter synaptic transmission at this overactive synapse. Did a number of biophysical studies in which we established the mechanism of this that I won't go into as a presynaptic inhibition of neurotransmitter release, but a very exciting effect in that we had identified a G protein coupled receptor, so a very druggable target, at a synapse that is thought to be a key synapse in this circuit that is defective in Parkinson's disease. So from that then we went to animal models to test the hypothesis that activation of that receptor would have an anti-Parkinsonian effect. The only molecule we had was this glutamate analog, AP4, so it was not a systemically active drug. We had to inject ICV, and this is a video that some of you in the audience have seen of a rat. This was the first rat that we performed this experiment on, in which we injected AP4 in the ventricles. This is a Parkinsonian rat that would sit in a completely immobile position for 24 hours if we did not inject the drug. You started to see these false starts as the drug comes on, and then this tremendous regain of motor function. The animal gets up, begins to walk normally, quickly ends the experiment, and it is ready to be through with science. So it was a very exciting effect. I remember when the people came from the lab to tell me this, they were calling it the Lazarus Effect because of this tremendous effect that they saw in these animals. And this was in the context of people who had been looking at a number of compounds for effects in Parkinsonian animals, and this was something that they clearly recognized as dramatic. But a very exciting illustration of going from clinical studies, dissecting apart the circuit, thinking about druggable targets, and then establishing in animal models that you can indeed have this activity. The problem with this target is that it has been very difficult to develop highly selective agonist of any of the metabotropic glutamate receptors. The glutamate binding site is highly conserved. LAP4 that I just showed you activates at least 3 subtypes of the mGluRs, but also is not really a drug lead. It could not be further developed because of various properties. And so there was a need for new compounds that could increase activity of this receptor, but not through interaction with the glutamate site. And this then goes to a point that Heidi made that has been a major focus of our lab now for many years, and that is thinking about ways to activate a receptor that is intractable through traditional approaches by targeting allosteric sites that might increase coupling of the receptor to G proteins and increase its activity. So thinking about the mGlu-Rs, these receptors have a large glutamate binding site in the extracellular domain, and then a 7-transmembrane spanning domain, and just before we began these studies, there had been success in identifying non-competitive antagonists of these receptors that bind to this extracellular region and inhibit receptor function but do not bind to the glutamate site. So we began to reason that we might be able to develop similar molecules that interact with this allosteric site. This was actually not mGlu-R4 but mGlu-R5, a different receptor. Now we're thinking of mGlu-R4, but trying to develop molecules that interact with that site and increase the ability of glutamate to activate the receptor. The only possible way to do this is a random screening approach because there was no structural information on the 7-transmembrane spanning domain of these receptors. We had no really reliable models to think about small molecules that could be designed through a rational approach. There were no molecules that had this type of activity, so we couldn't begin with known molecules and use molecular modeling at the small molecule level. So really all we were left with was a random screening approach to try to get a foot in the door and develop these molecules. And to do that, we developed an assay in which we expressed the receptor in CHO cells, looking now at calcium fluorescence. And the assay that we ran is shown here, where we add the test compound, wait 5 minutes, then add an EC20 concentration of glutamate, and we're looking for a compound that has no effect alone but potentiates the effect of this EC20 concentration of glutamate. This shows the first compound that we identified, a very dramatic effect, potentiating the effect of glutamate. Here you see a dose-response curve of glutamate alone and in the presence of increasing concentrations of this molecule, PHCCC, and we see a tremendous potentiation of activation of the receptor by the endogenous agonist. From this, we then performed selectivity studies and found that this compound was extremely selective and had no potentiator activity on any other mGlu-R subtype. Since that time, this compound has been screened through a large screen, pan-lab screen of several hundred different receptors, ion channels, transporters, and tends to be quite selective for the for mGlu-R4. So a real breakthrough in the first compound that selectively activates the single mGlu-R subtype. We then took this compound back to the studies that I just outlined, looked at the electrophysiology at the striatopalatal synapse, found that it does indeed potentiate mGlu-R4 actions there, and then in the animal models found robust efficacy. Once again, we had to inject PHCCC ICV because it was not a compound optimized for in vivo use, but with the ICV injection found a very robust anti-Parkinsonian effect similar to what we saw with glutamate. So this then provides the basis for a concerted effort to find new molecules that are drug-like molecules that could be further developed. We did a considerable amount of chemistry around PHCCC, and it was not really tractable as a drug lead, so we needed new molecules that had this activity. Here at Vanderbilt, we've now run a full high-throughput screen in the VICB HTS facility in which we have identified a large number of very robust allosteric potentiators of mGlu-R4. Some of these have nanomolar potency now. We're in the process of chemical optimization. Many of them have properties that make us think that they are going to be useful in vivo and ultimately useful as drug leads. So we're now in the process of a focused med-chem lead optimization effort that is progressing very nicely with a large number of hits to choose from that provide viable leads. The other thing that's nice about this project is, once again, we have some level of confidence of efficacy in the clinic, but it's not complete. You know, this still is a novel mechanism, and we may or may not see the type of efficacy that can be seen with the surgeries. But this is also an excellent program in that there is an animal model that thus far perfectly predicts clinical efficacy. So the first major go-no-go decision experiment for this program will be testing in the MPTP Parkinsonian monkey model. If that is successful, then we'll have a high level of confidence that we will see clinical efficacy. But the other advantage of this program is that it will be relatively simple to do early clinical development proof of concept studies to clearly establish that the efficacy will occur before going through this expensive development process on through phase 2 and phase 3. So we're excited about the progress of this program and continuing to move forward and making a lot of progress. The next program that I'll mention, I'm not going to take time to go through in detail based on our time, but I do want to at least mention it because it's a somewhat related but very different program, is an illustration of building on insights from clinical experience and serendipitous discoveries in the clinic where discoveries are made that give clear insights to novel approaches to treatment of disorders, but then we need new approaches that get around problems with those existing drugs. And this is a project focused on M1 muscarinic receptor activators for schizophrenia. So in this program, it began in the context of a major effort that we've had for many years, once again going back to my time at Emory and continuing at Merck and today, in which we have really been thinking about the different symptom clusters in schizophrenia without going into details about this, thinking of ways that we may develop approaches that could treat not only the positive symptoms but also negative and cognitive symptoms of this disorder. And a field that has emerged beginning in the 1950s that's been really exciting, and once again without going into detail for the sake of time, has led to the idea that manipulations that increase activity of a glutamate receptor called the NMDA receptor would have therapeutic efficacy in treatment of schizophrenia. The NMDA receptor is a ligand-gated channel. Activation of this receptor is critical for a number of aspects of cortical and limbic function and different systems that are critical for the pathophysiology of schizophrenia. And a fairly strong data set had emerged through clinical studies that supported this idea. So we had been studying different ways of modulating the NMDA receptor, and one that we found to be particularly interesting is that activation of the M1 acetylcholine receptor dramatically potentiates NMDA receptor function. This shows whole cell patch clamp recordings from neurons in the hippocampus. And what we noticed is that this occurs in all of the major neuronal populations that people would think could be important for increasing NMDA receptor function in schizophrenia patients. So this was a somewhat speculative idea, and certainly in my view does not fit clearly into this approach of really thinking critically about clinical studies. The M1 receptor is a step removed from NMDA receptor, but still an idea that we found of interest. But this occurred at about the same time that a very exciting discovery was made in the clinic from Eli Lilly, who had been developing a drug, xanomeline, for treatment of Alzheimer's disease. They took this compound all the way into phase 3 and found that it had no efficacy at the primary endpoint of cognition enhancement in Alzheimer's patients. But what they found is that it had a tremendous impact on what would be considered the psychotic symptoms that occur in schizophrenia patients, the hallucinations, delusions, paranoia, agitation, etc. And you can see here the effects on these different behavioral disturbances which were quite dramatic. So this was an exciting finding that dovetailed with the thinking about NMDA receptors and how muscarinic receptors regulate those receptors, and all of a sudden provided very clear clinical evidence that activation of muscarinic receptors would have therapeutic efficacy in schizophrenia. The thing that's amazing to me about this is that this discovery is so dramatic and clear, and yet it did not initiate a major activity of drug companies going after the muscarinic receptor for schizophrenia. What it did initiate was a lot of studies in, first of all, by Lilly, in one study in schizophrenia patients showing that it does indeed have efficacy in treatment of both negative and positive symptoms of schizophrenia patients, a small but very well-controlled, double-blind, placebo-controlled trial. And then a lot of animal studies showing that this compound has an antipsychotic-like profile in animal models. So these studies really fit well with the clinical data, but for the most part, most companies, including Lilly, were not that interested in the mechanism. And the reason was many companies had tried and failed to develop selective M1 agonists, all in the context of Alzheimer's disease, so that when this finding appeared, most companies and many in the academic community had really given up on the possibility of developing highly selective muscarinic receptor agonists. So we obviously were very interested in taking an approach of targeting allosteric sites, and we had such success with this with the metabotropic glutamate receptors that we were also interested in seeing if we could have the same success with the family A G protein-coupled receptor, M1 muscarinic receptor. And again, it was the same situation where we really had to start with what we had available. And what we began looking for were compounds that activate the muscarinic receptor but do not bind with high affinity to the orthosteric site. One of the compounds that came from this is this compound, TBPB. And we developed a system in which we looked at agonist activity. In this case, it's a direct agonist activity rather than allosteric potentiator activity. But what we found is that this compound activates the receptor at nanomolar potencies. It does not bind to the orthosteric site until you get up to close to 10 micromolar concentrations, so a clear disconnect between those. And we then made a mutation of the M1 muscarinic receptor that had been shown to eliminate activity of orthosteric ligands, such as acetylcholine. So this mutation prevents binding of acetylcholine, atropine, other known muscarinic agonists to the orthosteric site. It abolishes the ability of acetylcholine to activate the receptor. So this is a dose-response curve of acetylcholine alone and in this mutant. But for TBPB, the activation of the receptor is completely intact. So it's acting by a different mechanism. Through a number of studies, we've now shown that this is through binding to a site distinct from the orthosteric site. And then just to make a long story short and try to cut to the chase for the sake of time, like the allosteric potentiators of mGlu-Rs, this is a highly selective agonist. We've seen no activity at M2, 3, 4, or 5. It's a in vivo active compound that we can use for animal studies, enters the brain very well. We've now found that this compound has robust efficacy in multiple animal models that are used to predict antipsychotic activity. And we have the advantage of knowing the models where xanomeline is active, knowing that this activity for xanomeline did indeed predict clinical efficacy, so using those same models for this novel compound. But it has none of the peripheral side effects in these animal models that xanomeline and related compounds have. So once again, TBPB is a great tool, a great proof of concept compound, has allowed us to do a lot in terms of really developing an understanding of this target. But it is not really a drug lead for various reasons that we could talk about in individual discussions. So we have also been looking for new leads for M1 receptors. We've now completed a high-throughput screen here at Vanderbilt in which we've identified a large number of compounds, both with allosteric agonist activity like TBPB and also allosteric potentiators similar to what I showed you for mGlu-R4. And now we're pursuing these with chemical optimization. Once again, many of these compounds have all the properties that we would hope for in a drug-like molecule, and so now trying to develop these into authentic drug leads. I think I will skip that and just go right to the acknowledgments and thank a tremendous group of people that I am able to work with. This is the current makeup of my lab, which is just a very talented group of of individuals and collaborators. Those highlighted in yellow played major roles in the programs that we talked about today. Colleen Neiswinder really heads up the molecular pharmacology component of the lab, has played a major role in the mGluR4 work, but also M1. Carrie heads up our behavioral pharmacology activities. She has performed the behavioral studies with TBPB, also has done a lot with the Parkinson's models. Ashley, Brady, Joy, Marlowe, and Alice Rodriguez have all been involved in especially some of the high-throughput screening-related programs and then subsequent optimization. Jana Shiri and Jen Edel are both grad students in the lab that have each participated in the muscarinic side of this program. Jana has some tremendous compounds that I haven't shown you that are M4 selective allosteric potentiators, critical for that overall program. Carrie Myers, also a grad student who's been involved as a technician before she became a grad student. And then Mung and Cherry are both research assistants in the lab who have played a major role in these studies. None of this would be possible without the really talented groups, and this is an incomplete list in the HTS facility and different individuals here that are providing tremendous support in medicinal chemistry. Craig, Darren, Sam, Richard, Dave Weaver, of course, a key player in HTS. And then some of the studies that I referred to started at Emory with Guido Manioni and Michael Marino, and also continued at Merck with very important input from Cyril Sir and Gene Kinney. Have to always thank the people who fund the work, NIH, and in this case I am especially indebted to Vanderbilt, the Department of Pharmacology, the institution, the VICB, for the investment in the HTS facility, the chemical biology capabilities, all that is happening here in pharmacology, and none of this would be possible without that tremendous institutional support. So thank you very much, and I will stop there.
Questions for Dr. Conn?
Obviously, in a university study with Vanderbilt, you've got a good idea.
Mm-hmm.
But to move that along, you've got some money. You
That's right.
talk about risky things, preclinical, clinical, millions of dollars. What's your advice or your experience on acquiring capital to do those experiments?
Yeah, I really think about it in multiple stages, phases, and aspects. So one thing that I love about working in an academic environment is that the bar for success is a very different bar than in an industry environment, in that our goal, to me, when I see work that we do here at Vanderbilt have an impact to initiate a program in a company, even if we're not involved, that's a tremendous success. So for these programs, for example, I can now point to companies that have initiated programs based on this work where they are really trying to develop clinical development candidates and ultimately go into clinical testing. And in many cases, that's not necessarily in any direct collaboration with us, but once again, a tremendous success, I think, for Vanderbilt because we've been able to influence that. So that's part of the answer, is that we still have a primary academic mission and as biomedical scientists really focus on trying to improve health care ultimately through our basic science and a lower bar as to the parts of that we really have to personally contribute to. We're also very interested in being involved in going the distance, and I think to do that, it really takes partnering with companies and with others that have the capital to allow us. And it really depends project by project how far we should be able to consider taking things ourselves. You know, if we ever came to the point where we were able to get a compound that we could take through enough safety testing for an early proof of concept clinical study that was an experimental medicine hypothesis testing study, that would be terrific and would have a huge impact, I think, in really establishing efficacy. But there are a lot of other ways that could be done than trying to do that all internally. So I don't know that that's a really concrete answer, but for all the programs we're working on, I think of each of them differently. We've been very successful in getting the funding that allows us to do the types of things that I just told you about, and it's all in the basic NIH RO type and other NIH mechanisms. And I hope what you saw that there was a lot of this is basic science, and therefore it's perfectly suited to that. But if we were to lock in for full drug discovery, lead optimization, that then does become a different thing, and you have to think about different ways to get money for that.
Jeff, thank you very much for simulating our synapses. It was great. And I have two questions, but maybe first. 70% of failures due to low efficacy in central nervous system.
Right.
How much of it is due to the poor blood-brain barrier situation?
So I would.
You mentioned that your drug was getting nice to the brain.
Right. And so when I give that failure rate, that is failures of compounds that go into clinical development. So by the time you actually go into clinical development, you've spent a lot of time and effort and attention to thinking about blood-brain barrier penetration. And there are many, many discovery phase programs that never make it to clinical development because of an inability to get around that issue. But once you get around that issue and go into clinical development, you may have a situation where a compound, for everything you can do to predict blood-brain barrier penetration in all of the animal and other models you have, is not adequate, and you go into men, and that's an issue. Total pharmacokinetic issues, blood-brain barrier would just be one of those, is on the order of 20% of failures. So, or actually, no, it's more like 10%, then 20% is for toxicity and other things. But usually the pharmacokinetics today, we've become much better at predicting human pharmacokinetics from the animal studies, and so it's not the same problem it was 10 years ago or so.
Now, your 14 years time for development of drugs includes about 2 to 4 years target identification.
That's right.
But those are small molecules.
Right.
Now, we have monoclonal antibodies, which can be developed for any target,
right.
especially in And what's your kind of view of 350 different monoclonal antibodies that are now in the pipeline?
Yeah, I mean, I think biologicals in general, monoclonal antibodies, have tremendous promise. That we've seen major breakthroughs just recently, and that even includes neuroscience. So it's not just restricted to therapeutic areas that you would really think of with monoclonal antibodies. So to me, it's very exciting. I see it as a tremendous opportunity, tremendous promise. The issues with drug discovery and development for biologicals, including monoclonal antibodies, are very different from small molecules. And the failure rate there is, predicting efficacy is oftentimes easier. One of the big issues is production. So when you look at the total cost of drug discovery and development versus production for small molecules, it's all on the discovery and development side. Once you have a small molecule, production is usually relatively minor. But in biologicals, there are cases in which the efficacy is there, everything is there to go forward, but the hurdles with production become insurmountable. So it's just very different issues for the two. Both of which I think offer a great promise that we should all be excited about.
We took the idealistic position that the goal of drug development is to find those medications that are essential for caring for human illnesses which cannot be prevented or altered in any other way. So we take that position. Would we be better off without a patent system or by altering the patent system?
I think there has to be an incentive for doing this in the business world. So from an academic position and the things that we can bring to bear on that whole process here, it's a very different set of motivators and drivers. But ultimately, for drugs to make it all the way to the point that they are on the market and being used in the clinic, there have to be incentives. So there has to be a very strong patent system and patent laws that are enforceable and followed in order to allow that to happen. And I hope one thing that you saw is this huge investment is an extremely risky venture, and even if we do our job of trying to reduce that risk, increase the success, it's always going to be a very costly venture, and there has to be an incentive, I think, in the business world to pursue those studies to get medicines. So one thing that I would fear is that it becomes so onerous and so difficult and costly. The incentives are diminished to the point that there are no major efforts at developing medicine. So I think we have to have a strong patent system. What the best patent system is to facilitate that, I don't know. But so I would think between those two options, it would be altering our current patent system.
I know there are a number of other questions, but I'm going to ask you to please talk with Jeff later because there's a reception outside, and I think we've gotten to the hour that we would like to get there. So thank you again, Jeff.
Thank you.
For a really